Hello, welcome to Sense of School of Life Sciences. This video is about glycogenesis. Glycogenesis is the synthesis of glycogen from glucose. This occurs in all the tissues of the body, but the major sites are liver and muscles. Now see the different steps. First of all, glucose is phosphorylated to glucose 6-phosphate by the enzyme glucokinase or hexokinase in the presence of ATP. Hexokinase and glucokinase are isoenzymes, that is, different enzymes that catalyze the same reaction. Hexokinase is present in almost all tissues, including muscles, while glucokinase is present only in the hepatocytes of liver and beta cells of pancreas. Glucose 6-phosphate is converted to glucose and phosphate, catalyzed by the enzyme phosphogluconeosidase. Then glucose and phosphate is converted to UDP glucose, which is catalyzed by UDP glucose pyrophosphorylase. It requires UTP. Remember, UDP glucose is the activated glucose donor in the process of glycogenesis. Then this UDPG can be used for the synthesis of glycogen with the help of glycogen synthase enzyme, which is the rate-limiting enzyme. But the glycogen synthase cannot start the synthesis of glycogen on its own, but it requires a primer. Remember, glycogen synthase to function, a primer is required. So how the primer is synthesized? For the synthesis of glycogen primer, we require a molecule known as glycogenin, which is a dimer protein. It's a glycogenin. A glucose molecule from UDPG is transferred to the glycogenin protein. I told you, UDPG is the activated glucose donor in the process of glycogenesis. This reaction is catalyzed by glycogen protein itself. That is, it is autocatalytic activity. Glycogenin protein, you see, one glucose is added, then further glucose molecule up to 8 glucose molecules will be added by the extension activity of this glycogenin protein itself. So then it is further extended by other glucose molecule from the UDPG itself. This activity is also catalyzed by glycogen itself, that is autocatalytic. Thus, glycogen act not only as a primer, but also act as an enzyme. Thus, 8 glucose unit long chain is formed. This is the glycogen primer, which can act as a primer for glycogen synthase. Then, lengthening of the chain. Glycogen synthase enzyme transfers the glucose from UDPG onto the non-reducing end of glycogen primer. So non-reducing end. The glucose molecules are linked by alpha 1,4 linkage. It is linked by alpha 1,4 linkage. See, 1,4 linkage. There. Then we can see the branching process. Branching occurs with the help of branching enzyme. But when the chain of glycogen molecule have been lengthened up to a minimum of 11 glucose residue, or it may be up to 12 or 13, or here it is up to 14. So when the chain of glycogen molecule have been lengthened up to a minimum of 11 glucose residue, the branching enzyme remove a segment of about 6 glucose residue. You see here 9, 10, 11, 12, 13, 14, about 6 glucose residues, and reattach this fragment on the original chain by the which linkage? Alpha 1,6 linkage. Alpha 1,6 linkage here. Linkage to form a branch. The branching enzyme deficiency may cause Anderson disease also, that also remember. Okay. Thank you.